Hello everyone and welcome to Chapter 15, Genes and Proteins. This is the first lecture in a series of 2. So I love this image. This is an image of erythrocytes, red blood cells. This is an actual micrograph of red blood cells. This is what they look like. They kind of look like little donuts, little jelly donuts, right, that kind of have been pressed in the middle, but the hole doesn't go all the way through. The neat thing about this is that about 96% of the content of red blood cells is protein. This chapter is about genes and protein, so I wanted to point that out. And most of that protein is hemoglobin. Now you've kind of been introduced to hemoglobin back when we were talking about the levels of protein structure, right? And hemoglobin, if you recall, is a protein representing quaternary structure. So you'll see that hemoglobin, it's huge, and it's composed of 1, 2, 3, 4 separate protein units. Okay, and these units, each of these 4 units, is in tertiary structure. It's a tertiary level of protein structure. And then if you'll recall, in order to get to quaternary structure, you take a bunch of tertiary structured proteins and you lump them all together. So these are all connected. These are all tertiary structures, all connected. There's 4 of them, so that makes quaternary structure, and these work together. You can see these little iron molecules here. Each one of the 4 units has an iron molecule, and it's the iron that carries oxygen in the red blood cell, the oxygen that gets to your muscles and helps your tissue stay alive. Okay, so I just want to point that out, that most of the protein, most of the structure, most of the internal contents of red blood cells is protein, and most of that protein is hemoglobin. And that's what we're talking about. We're talking about in this chapter in making proteins. So at this point in your Biology 101 journey, you know what genes are, you know what they're made of, you know how genes determine specific physical, remember that's phenotypic characteristics, and how those traits can be transmitted between generations. So now we want to see how the information, that is the genetic information that determines your physical traits. So we want to see how the genome or the genotype gets to the phenotype. So we want to find out how the genetic information that determines physical traits gets from your DNA to your hair color or your eye color or your height or your blood type or 5 fingers on each hand and 6 toes on each foot, or whatever your physical characteristics are. Okay. So if DNA is analogous to the blueprint for the house in this image, right? All buildings have a blueprint. So if DNA is the blueprint for the house, or analogous to the blueprint for the house, and the house is the physical expression of the blueprint, so this is the phenotype, then you are the physical expression of your DNA blueprint, right? So you are the house when it comes to your DNA blueprint. You are analogous to the house, and the process of translating the blueprint into the house is the subject of this chapter. So your objectives are to distinguish between transcription and translation, describe the genetic code, which you've already been introduced to, explain the relationship between codons and amino acids, differentiate initiation, elongation, and termination of transcription, identify the unique features of prokaryotic transcription, describe the purpose of promoters, describe the processing of transcripts, explain the relationship between genes and proteins, explain the purpose of transfer RNA, that's the little t, capital RNA, that's transfer RNA in translation, and describe the nature of genetic mutations. We'll look at all of this in Chapter 15. So again, I have said that you have been already introduced to the central dogma via this image right here. Now, you know all about replication. Replication occurs during the synthesis phase of interphase of mitosis, right? And that's where DNA is replicated, right? You go from one chromosome to a homologous chromosome, right? You copy it. It's like a Xerox copy of your DNA. That's replication. But the central dogma is DNA to RNA to proteins. You'll notice that information flows in one direction. You can't go back. And so you're going from DNA, which is genetic information, the process called transcription, which we'll learn about, to RNA, which is genetic information, and from RNA, the process called translation, to protein. Protein now is phenotypic information. So we go from genetic to phenotype, or genotype to phenotype. And so we're going to look at these two processes, transcription and translation. That's the central dogma. So replication, you are making DNA from DNA. So you're going from double-stranded DNA to double-stranded DNA. It's DNA-directed synthesis of DNA. We have to remember these enzymes. DNA polymerase makes DNA from DNA. And the two strands in replication are complementary. Remember, complementary base pairing. Okay, transcription then is DNA to RNA. So you're going from DNA, which is a double-stranded molecule, macromolecule, to RNA, which is a single-stranded macromolecule. Okay, this produces an exact copy of DNA, which is double-stranded, as a single strand of RNA. And we call that the message. That's the little m, stands for messenger RNA. So you've already been introduced to transfer RNA and the objectives, and now we're being introduced to messenger RNA. And you'll remember that terminology as we get into the processes. So not unlike transcription of a trial, which contains the exact words of a court proceeding, right? So you're in court, you've seen this on TV. If you hopefully haven't experienced this for real, but there's always a person who is typing up every word that the defendant says, that the prosecutor says, that the judge says. Every single word of that trial, court proceeding, is written down, is recorded. That process is called, or that person is called, the transcriptionist. And that exact word-for-word record of that court proceeding is called the transcription. All right, so you see where that comes from? So what we're doing in transcription is we're making an exact copy in RNA of DNA. So it's like in a court proceeding, DNA is like the verbiage, right? It's what everybody is saying. So you're going from the words that everybody is spitting out to the written record of those words, and that would be your messenger RNA. Transcription is a DNA-directed synthesis of RNA. Now, here's the trick with polymerases. DNA polymerase, right, catalyzes the replication of DNA to DNA. Transcription is DNA-directed synthesis of RNA. However, the enzyme that does the work is RNA polymerase, right? So DNA polymerase makes DNA, RNA polymerase makes RNA, even though RNA polymerase is directed by DNA, all right? And the single strand of RNA is complementary to the strand of DNA that acts as, we call it the template strand or template strand. Okay, and then we get to the process of translation, and that's where we go from RNA to protein. So here is the actual process where we go from genotype to phenotype. Remember, protein is the physical structure, right? Your hair is made of protein. Your nails are made of protein. Your skin is made of protein. So the phenotype, your phenotype, whatever we see, those characteristics that I see when I look at you or that you see when you look at me, that's all directed by protein or all substantive of protein. Okay, so translation then translates the language of nucleic acids into the language of proteins. That's why we call it translation. Okay, so let's go to the video. It's called The Central Dogma of Biology. It's from the DNA Learning Center. Here's the link. I have the video embedded in your notes. Check it out. It's a nice little descriptive animation of the central dogma of biology to help you better understand that. And then here is just a little fun presentation of the central dogma. It's called The Central Dogma Song Sing-Along by Kev Is Gone. And you can watch it. It's kind of funny. You can watch it if you want. Okay, so the two processes are transcription and translation. Here we're looking at transcription. Now, you want to be able to do this. You want to be able to take a strand of DNA and transcribe a message from there. Okay, so DNA is double-stranded, while RNA is single-stranded, so only one strand of DNA is needed for transcription. We call that the template strand or template strand, right? The template strand is used to make messenger RNA, there it is again, and the other strand is the coding strand, okay? Now, this does not need to be complicated, as we'll see, all right? So stay with me. Transcription uses complementary base pairing, right? So just as you've learned from the lab and from the and from Chapter 14, DNA Structure and Function, the two strands of DNA, we say they're complementary, right? So T pairs with A, G pairs with C, okay? Now, in order to transcribe the message, we need to tease the two strands apart. We need to pull them apart of DNA, I mean, because remember, it's double-stranded. It's the double helix. It's like the twisted ladder. So we need to untwist it, and we need to pull the two apart temporarily. This is not a permanent situation. We pull the two apart, and then we can use one strand to make the complementary strand of RNA. Remember, RNA is single-stranded. Okay, so it's sort of like we're making a mirror image of DNA, but we're making it in RNA. How do you tell the difference? Because of the bases, okay? So because of the nucleotides. So in RNA, so you know that with DNA you have A, T, C, and G, right? In RNA, you replace the T, the thymine, with uracil. So in DNA, we have adenine pairing with thymine. How can you tell the difference between RNA and DNA then? Well, you remove thymine in the DNA, or in RNA really, and you replace it with uracil. So if you see a single strand of nucleotides and U is in there, you know that's RNA. If you see a single strand of nucleotides and it's A, T, C, and G, you know that's DNA. If it's A, U, C, and G, you know that's RNA. So the coding strand then, we call it the sense strand or the plus strand or the positive strand because it has, we call it the sense strand because it has the same sense as the message. That's the coding strand. Well, this will make sense in a minute. And the template strand, we call that the antisense or the negative or the minus strand. It's anti to the sense strand. So what you're going to do is you're going to take a double strand of DNA and you're going to unwind it and you're going to pull it apart. One of those strands is going to be the coding strand or the sense strand, and the other strand is going to be the template or the antisense strand. So here is your double strand. Now this is double-stranded DNA right here. Okay, there it is, double-stranded DNA. Now we've untwisted it until we have our two lengths of DNA. This is the sense strand here, 5' to 3'. And here's your template strand, 3' to 5'. We call it the antisense strand because it's the opposite of the sense strand, right? So in the sense strand you have TCA, right? So in the template strand, the antisense strand, is the complement to TCA, which is going to be AGT. So there you see AGT, and you can do that all the way down. GCC, that's going to be CGG. GTC, that's going to be CAG. Okay, you do that all the way down, right? So this is the antisense is the opposite of the sense strand. Now, when we make the message, we're going to use the antisense strand because we want to make a complementary strand of RNA, a complementary strand to this strand, to the sense strand. Does that make sense? It should. All right, so in order to make a complementary strand to the sense strand, we have to use the antisense strand. We have to use the opposite side. And so that you know that in RNA you're going to replace T with U. So everywhere you see a T, you're going to replace it with U. Okay? Or I should say, everywhere you see an A, you're going to replace it with U. Okay, so here is the message, the messenger RNA. Okay, now, okay, I'll say that in a minute. Okay, so we have AGT. Now normally that's going to pair with TCA, but we don't have any T's in RNA. So A pairs with U in RNA. G still pairs with C, T still pairs with A, C pairs with G, G pairs with C, G pairs with C, C pairs with G, A pairs with uracil, because we don't have any T's in RNA. G pairs with C, T pairs with A. Okay, see how that works? So what happens is, all right, and so this is your message, the messenger RNA. You know it's RNA because it has U in it. And so what happens is this strand, 5' to 3', is the same as this strand, 5' to 3', except wherever you see a T, you replace it with a U. So that's how you know when you do this exercise, and you'll do a number of these exercises, and certainly you do one on the test. That's how you know you are correct. When your message matches up, it's exactly like the sense strand or the coding strand of your DNA, except T is replaced with U. Right? So if you went back and you looked at it, right, and if you were thinking of this as DNA, this would be TCA, GCC, GTC, like that. See? TCA, GCC, GTC. It's the same information here as up here. The same information. That's how you know you're correct when you do your message, when you transcribe your message. That's how you know you're right. If it's exactly the same as a coding strand, except you have U's where you have T's, or you replace the T's with U's, then you know you're right. Okay, so that's the message. So we use the antisense strand to make the complementary RNA to the sense strand. That's transcription. You are transcribing the DNA into messenger RNA. Okay, so transcription is DNA to messenger RNA. Translation, then you take that message, then if you watch the video, you see it gets so all of the transcription, replication, transcription takes place inside the nucleus. Translation takes place outside of the nucleus. So you got to get the messenger RNA outside of the nucleus. How do you do that? Well, you're familiar with the structure of the nucleus, and the nuclear membrane has all of these pores in it. So the messenger RNA is thin enough to fit through those pores. So it goes out through the pore, out of the nucleus through the pore, and then the ribosomes can connect to the message and then can translate it. So translation occurs on the ribosomes, and it requires several types of RNA, and we've already talked about messenger RNA. We made that through transcription. We've already talked about transfer RNA, and the other RNA is the ribosomal RNA, which is lowercase r, capital RNA. So the messenger RNA, that's lowercase m RNA, carries instructions for the polypeptide chain to the ribosomes. This is what we just made when we transcribed the DNA. We made the message. The ribosomal RNA, that's lowercase r RNA, serves as a major component of ribosomes, and it's the ribosomal RNA via the ribosomes that come together that are able then to read the message and translate it into protein. And then you have transfer RNA, which is lowercase t RNA, carries the amino acids or the proper amino acids to the ribosome as the message is translated. Here you see some illustrations representing the different shapes of the message. Single-stranded RNA. Here's ribosomal RNA inside of the ribosomes. Ribosomes are made up of two components here. And there's your transfer RNA, which kind of has this funky shape to it, but what you will notice is, these are nucleotides on your message. These are nucleotides on your transfer RNA, and that's really important as we get into the actual translation. Okay, let's go to the video from DNA to protein. It's a 3D video by Your Genome. It's really good. It kind of goes fast, but these are the things that are happening via translation in real time. It's incredible how fast the process goes. It's real time, and it's 3D animation, so it's really, really cool. You might have to watch it a couple times to kind of get the full impact of the process, but just keep in mind as you watch it, this is in real time. This is happening in your body right now. If you have any questions, make sure you let me know via email message or the Canvas inbox, and that will be the end of the first lecture from Chapter 15.